Hello, my name is Marion Bornhorst, and today's date is August 27th, 2019. And today we'll be discussing case connections for Unit 2, and I will be going over case connection 2 and 3. So, case 2 presents us with a topic based on how glucose uptake is regulated by the cell in different tissues, and the case focuses on a glioblastoma tumor in the brain that's depicted both by MRI and PET scan. And the case wants us to determine how or if the glucose uptake differs in a tumor as compared to other tissue types, but it is important to review how other tissue types uptake glucose before we can make that comparison. So the major tissues we will be discussing include the liver, skeletal muscle, brain, and adipose tissue. All of these tissues utilize glucose in the fed state to provide energy for underlying metabolic processes. However, the way in which glucose enters all those cells varies. So to begin, we'll discuss adipose and skeletal muscle, which uptake glucose through a primary glucose transporter named GLUT4. And this glucose transporter is controlled primarily by the hormone insulin, which is secreted by the beta isolate cells of the pancreas in response to an increased concentration of glucose in the blood. So GLUT4, unlike other primary glucose transporters, is intracellular, and an early response to insulin migrates those GLUT4 vesicles to the cell surface, where they can fuse with the plasma membrane, exposing the active glucose transporters and allowing for glucose uptake. So this system of using GLUT4 allows for glucose uptake to be regulated by insulin, and in the fasting state, the skeletal muscle and adipose tissue are taking up less glucose because there is less insulin. So focusing in on the liver, it takes up glucose in an insulin-independent manner. So it uses the glucose transporter GLUT2, and this transporter is always present on the cellular membrane. Insulin, however, is thought to indirectly promote uptake in the liver by encouraging the synthesis and storage of glucose to glycogen. However, insulin doesn't directly affect glucose uptake by the liver. And the last tissue that we're going to focus on is the brain, which is the tissue that is shown in the images for this case connection. And just as the liver, the brain takes up glucose in an insulin-independent manner. And in this case, the brain uses GLUT transporters GLUT1 and GLUT3 to transport glucose into the cells. GLUT1 specifically transports glucose across the blood-brain barrier, and GLUT3 controls the uptake of glucose into the neuron. So overall, with brain metabolism, the brain will always preferentially oxidize glucose, except in the case of extreme starvation, where the brain can use ketone bodies that are produced by the liver in a fasted state. So after reviewing glucose uptake in major body tissues, it's time to answer the question, how does glucose uptake in normal tissue compared to glucose uptake in the tumor cells? So this question is simply shown in the PET scan. Glucose uptake by cancer cells is significantly increased compared to normal tissue, and this is advantageous because it allows for fueling of all metabolic processes, which allow cancer cells to grow and divide at a quicker rate. And the last question is what modifications may have occurred in cancer cells to alter glucose uptake as predicted above. So I found a scientific journal that I thought was really interesting, named The Biology of Glucose Metabolization in Cancer Cells. And the authors discussed glycolytic enzymes such as GLUT1, lactate dehydrogenase, and pyruvate kinase as being unregulated in cancer cells. Which this is important because these enzymes being unregulated leads to an increased glycolytic flux for the tumor cells. And then another piece of evidence from that same article that I found interesting was how cancer cells switch from a cellular respiration to a primarily glycolytic pathway to produce ATP. And this switch is known as the Wahlberg effect. And the reason that this switch is still under speculation, but it's a means of metabolism that is thought to produce metabolic end products to support rapid growth in cancerous cells, as well as the ability to proliferate under low oxygen tension because it's not using an electron transport chain. So even though this seems like a highly inefficient way of producing energy, the cells make up for this energy demand by going through glycolysis much faster than a normal cell would, meaning that these cells need to absorb more glucose in the first place. So by solely using glycolysis as a means of ATP, the cancer cells must adopt a mode of increased glucose import to meet energy demands. So this leads to the overexpression of GLUT1 and GLUT3 glucose transporters, as discussed before, as well as an overexpression of the glycolytic enzymes. So that wraps up case 2, and then I will discuss case 3. And this kind of relates to insulin in a different way. It gives us serum insulin levels at baseline in 1 hour after glucose load of a 5-year-old boy who comes in for an annual checkup to his doctor. And the case wants us to put this information with the symptoms that he's reporting to predict what is going on in a biochemical sense. So the main symptoms that are important in this case are drowsiness, persistent thirst, and frequent urination. So in hearing these symptoms, I first think of hyperglycemia because these are common symptoms of individuals with continuously high glucose levels. And as we know, hyperglycemia is often found in diabetic patients. And the diagnosis would also make sense based on the individual's serum insulin levels as shown in the case connection. And at baseline, the patient's insulin level is 0.8 milliunits per liter as compared to the control, which is 6 plus or minus 2 milliunits per liter, which is a huge difference. And then even following the glucose the patient's insulin level is far below the control too. It presents as 5 milliunits per liter as compared to the control, which is 40 plus or minus 11 milliunits per liter. So these low levels of insulin in a patient his age normally arise from type 1 diabetes, which is where the beta cells of the pancreas genetically don't produce enough insulin because these cells are being attacked by the immune system and causing apoptosis of these cells. So without proper insulin levels, glucose will accumulate in the blood because cells don't have that hormone signal to take up glucose, which is needed. So the next question to touch on asks us, what would you predict the patient's fasted blood glucose levels to be given his current situation? So in this scenario, the boy lacks enough insulin to take glucose out of the blood after a glucose load test. So this means that insulin present will allow for an uptake of glucose at a much slower rate than in a normal fed state. So in the fasted state, we can predict that the patient's glucose levels would still remain above normal range, as the insulin can't keep up with the glucose levels of the blood, and it also can't effectively signal for cells to take up glucose, so glucose would remain high in the fasted state. So lastly, we will touch on the regulatory enzymes that I would predict in this patient's condition. So this patient's condition relates to low amounts of insulin, leading to low levels of glucose uptake and high levels of glucose in the blood. So at the cellular level, this means that the cell wants to promote glycolysis to produce ATP from the glucose that it is able to uptake, as well as wants to suppress gluconeogenesis so that more glucose is not released into the blood. So 3 regulatory enzymes in the promotion of glycolysis are glucokinase, also referred to as hexokinase, phosphofructokinase 1, and pyruvate kinase. And I expect all of these enzymes to be highly active in this patient in the promotion of glycolysis. And glucokinase is responsible for the conversion of glucose to glucose 6-phosphate in the liver in the presence of high levels of glucose, which we see in this condition because the patient has elevated glucose levels in the blood. PFK1 is also is also responsible for a major part of glycolysis by phosphorylating fructose 6 kinase to fructose 1,6 bisphosphate. And then finally, last enzyme that I mentioned that could be regulatory in his condition is pyruvate kinase, which converts phosphoenol pyruvate to pyruvate that can then enter into the citric acid cycle. So I expect all of these to be elevated. And that concludes the second and third case. So thank you very much. Have a great day.